Okay, welcome to part 2 of the Mod 13 lecture on genetics. We're going to pick up right where we left off. We finished up talking about monohybrid crosses. In this second part of the video, we're going to talk more about Mendel's laws and talk specifically about dihybrid crosses. So a monohybrid cross is a cross between two individuals that are heterozygous for just one character. The example we used was flower color. A dihybrid cross is a cross between two organisms that are each heterozygous for two characters that are being followed. So an example of that, flower color could still be one. The other one might be seed texture, whether it's smooth or wrinkly. Mendel's law of independent assortment states that the alleles of a pair segregate independently of other allele pairs during gametes formation. So this means that the alleles for flower color segregate independently of the alleles for seed texture. And so flower color has no influence on what texture of seed you will get. If that's still a little bit confusing, we're going to work through a few examples. This here shows a cross just focused on two factors, but we're no longer looking at flower color. We're looking at seed color and then seed texture. So those are our two factors that make it a dihybrid cross. We have color, can be yellow or green, and then texture, which can be smooth or wrinkled. On the left here is what would happen if assortment was dependent. So if the smooth seed could only be inherited with the yellow color, and if the wrinkled seed texture could only be inherited with the green color, we would call that dependent assortment because the alleles are not independent of each other. Smooth and yellow go together, and then wrinkled and green would go together. If that were the case, if you crossed a smooth yellow seed with a green wrinkly seed, you would see that the offspring in the F1 generation would all be RRYY, capital R, lowercase r, capital Y, lowercase y. They would all be yellow smooth. And then in the F2 generation, we would see that the only possible traits are the traits that we saw in the parents. So the seeds can either be yellow and smooth, or they can be green and wrinkly. You cannot have a yellow wrinkly seed or a green smooth seed. Hopefully that's making sense. So the hypothesis of dependent assortment is that the seed texture and the seed color are inherited together. The alleles are inherited together. So these are the results we would expect to see if that was the case. However, you probably noticed the big yellow circle with the slash through it, indicating that this is not what Mendel found. So this is not what happened. His hypothesis was refuted. What he actually found was over here, which supported his hypothesis of independent assortment. I'm going to talk through this one a little bit more too. So again, we have the P generation up here. The yellow smooth seed is homozygous dominant for both color and for smooth or wrinkly. In this case, we'll say that R is the color and Y is texture. So capital R is yellow, capital Y is smooth. And then, sorry, I did not mean to do that. Over here on the green one, lowercase r is green, and then lowercase y is wrinkly. Both individuals in the P generation are homozygous dominant on the left, homozygous recessive on the right. So there's only a limited amount of gametes that they can pass to their offspring because that's all they have. The green wrinkled seed only has lowercase r's and lowercase y's. And then the yellow smooth seed only has capital R's and capital Y's. So those are the only gametes it can pass on. As a result of this, all the individuals in the F1 generation will be heterozygotes. So they will have a capital R from this parent, a lowercase r from this parent, a capital Y from this parent, and then a lowercase y from this parent. Our F1 generation are all heterozygotes. Then we're going to cross that F1 generation. So R, lowercase r, capital Y, lowercase y. We're going to cross that with another individual with the exact same genotype. These are the results that Mendel actually found. So the alleles assort independently, and we can see that not only do we have yellow round seeds, which were one of the parents, and green wrinkly seeds, which the other parent was, we also have yellow wrinkly seeds, which we did not see in the parent generation, and green round seeds, which we also did not see in the parent generation. Based off of this, Mendel concluded that the alleles assort independently, and so seed color is inherited independently of seed texture, which is how we can end up with offspring in the F2 generation that don't resemble either parent, such as the yellow but wrinkly and then the green but round. This Punnett square, I know there's a lot going on. I'm not going to go through how to set this one up. On the next slide, we have an example that I'll actually walk you through how to set one of these dihybrid cross Punnett squares up. So in our example, we are going to use Labrador retrievers. We're not going to deal with the yellow lab color because that's actually a completely different gene. We're going to deal with the coat color gene that controls black lab or chocolate lab. And then we're going to deal with the gene that controls normal vision or the development of a disease called PRA or progressive retinal atrophy, which causes the dog to go blind. These are all the different genotypes that black and chocolate labs can have. The black color is the capital B. It's the dominant coat color allele. And normal vision, so no progressive retinal atrophy, is the dominant allele for vision. So the black coat is represented with a capital B. Normal vision is represented with a capital N. If a dog is blind, if it has PRA, progressive retinal atrophy, that means it's a homozygous recessive for the vision genes, which are lowercase n, lowercase n. And then if a dog has a chocolate coat, chocolate is the recessive, so the chocolate coat will be 2 lowercase b's or homozygous recessive for the coat color. Since we're looking at 2 traits, there's 4 possible phenotypes. We can have a black coat with normal vision, so that just means the individual will have a capital B and a capital N because the capital B is the dominant trait for black color. Capital N is the dominant trait for normal vision. We can have a dog that has a black coat, which is the dominant trait, so they'll have a capital B, but they're blind. So because they're blind, we know that they're homozygous recessive. They have the 2 recessive alleles for vision, the lowercase n, lowercase n. We can have a dog that has a chocolate coat, which is the homozygous recessive allele for coat color, the 2 lowercase b's, and normal vision. Normal vision is the dominant trait for vision, so it just means they have 1 capital N. And then finally, we can have the fully homozygous recessive individual that is blind and has a chocolate coat. So the chocolate coat, again, is the recessive coat color, so lowercase b, and then blindness is the recessive vision trait, so that's lowercase n, lowercase n. We are going to mate double heterozygotes that have black coat and normal vision. So those double heterozygotes, heterozygote means they have both dominant and recessive. So if they have a black coat and normal vision, we know that their genotypes are capital B, lowercase b, capital N, lowercase n. And then we're going to cross that with an individual of the exact same genotype of capital B, lowercase b, capital N, lowercase n. This down here is the actual results from the cross. Don't pay attention to that right now. I'm going to go ahead and show you how to set this up on the whiteboard. So we have our dihybrid cross for coat color and vision in labs. So we're crossing 2 heterozygotes are the parents. So we have a capital B, a lowercase b, capital N, lowercase n, that's one parent. We're crossing it with another parent that has the exact same genotype that is heterozygous for both traits. So we have capital B, lowercase b, capital N, lowercase n, crossed with an individual with the exact same genotype. Now we have to figure out how to put these traits onto this Punnett square. This is where it gets a little tricky. Since the alleles assort independently, we need to put all the possible allele options. So starting with this parent over here. We're going to put this parent down the left side. Each box is going to have a letter for coat color and a letter for vision. Each box going down. So there'll be 2 letters here and then 2 letters up here. Starting with this parent, we're going to just go from left to right. So we're going to start with capital B. And now we know it needs a vision allele, so we're going to do capital N. Now, there's another possible arrangement with a capital B, so we're going to do the capital B again. And this time, we're going to do capital B with the lowercase n. So these are the 2 possible options for alleles with the capital B. Now we're going to switch to the lowercase b. So it will be lowercase b and then capital N and then lowercase b again and lowercase n. These are the allele options for this parent and they're listed out here. Now we're going to do the exact same thing for this parent, starting with the capital B. So the first option is capital B, capital N. So that and that. The second option is capital B, lowercase n. Now those are both of our options for the capital B. Then we have a lowercase b and a capital N. And then we have a lowercase b and a lowercase n. So we have now set up the Punnett square correctly. Now we need to go ahead and fill it out. Each box will have 4 alleles inside of it. So to do this first box, we're just going to do the B's first and then follow it by the N. So we have 2 capital B's and then 2 capital N's. To do this box, we have 2 capital B's again. And I'm just going to list the B's first for clarity. We have a capital N and then a lowercase n. So now we have a capital B, a lowercase b, and then 2 capital N's. And then finally we have a capital B, a lowercase b, and then a capital N and a lowercase n. So that is the first row. I'm going to go ahead and fill out the rest of the rows with just a little bit less explanation. So this row will be capital B, capital B, capital N, lowercase n. This one will be capital B with 2 lowercase n's. This one will be capital B, lowercase b, capital N, lowercase n, capital B, lowercase b, and then 2 lowercase n's. This row will be capital B, lowercase b, and then 2 capital N's. This row will be capital B, lowercase b, and then capital N, lowercase n. Here we have 2 lowercase b's and then 2 capital N's. Notice that I'm always listing the b's first. And then here we have 2 lowercase b's, 1 capital N, and a lowercase n. Here we have a capital B, a lowercase b, and then a capital N, and then a lowercase n. Capital B, lowercase b, and then 2 lowercase n's, lowercase b, capital N, lowercase n, and then finally lowercase b, lowercase n, lowercase n. Notice that I always write the capital letter first. That's just a general accepted way to write alleles, and genotypes is to always write the dominant first followed by the recessive. So I made sure the first rule was that B's always came before N's, and then after that I would always write, if there was a capital letter, the capital letter would come first before the lowercase letter. That's all I was doing. So we have just filled out our first dihybrid cross Punnett square with 16 squares. You can look at the genotype ratio of a dihybrid cross. It is a lot to look at that, and I'm not going to ask you to do that. What I will expect you to be able to do is to determine the phenotypic ratio from a cross like this. So the phenotypic ratio, remember, is the physical traits. And so I want to know how many of the labs have black coats and normal vision, how many of the labs have black coats but are blind, how many are chocolate with normal vision, and then how many are chocolate and blind. So we can quickly tally this up. We're first going to tally up black labs with normal vision, so they need to have a capital B and a capital N, at least one of each. It looks like we have 1, 2, 3, 4, 5, 6, 7, 8, and 9 right there. So we have, I'm going to change the color, 9 black labs with normal vision. The criteria for counting the 9 was they had to have a capital B and they had to have a capital N. The next phenotype is black coat but blind. So this time we're looking for genotypes with a capital B but only lowercase n's. So they have blind, not normal vision. They are blind. So a capital B and only lowercase n's, 1, 2, and 3 right there. So we have 3 offspring that are blind and have black coats. Now we're going to look for chocolate coats normal vision. So 2 lowercase b's and a capital N. Let's see, we have 1, 2, 3 of those. And then finally, we're looking for the homozygous recessive, all lowercase letters. We only have one of those right there. This gives us a ratio of 9 to 3 to 3 to 1. This is the typical phenotypic ratio when you're doing a dihybrid cross. Again, I won't ask you to calculate the genotypic ratio of the different genotypes, but I will expect you to know how to calculate these phenotypic ratios. We have 9 black labs with normal vision, 3 black labs that are blind, 3 chocolate labs with normal vision, and 1 chocolate lab that is blind. This is our phenotypic ratio. Going back to the PowerPoint, you'll see that they have the same results here. They had 9 to 3 to 3 to 1 for the phenotypic ratio of the offspring. 9 black coat normal vision, 3 black coat but blind with PRA, 3 chocolate coat with normal vision, and then 1 chocolate coat that is blind. So that was an example of how to calculate out a dihybrid cross. Now a question based on this. Hopefully you didn't see the answer because I know this slide was up when I went to the whiteboard. Predict the phenotypes of offspring obtained by mating a black lab homozygous for both coat color and normal eyes with a chocolate lab that is blind from PRA. So first I want to talk about this chocolate lab that is blind. We know right away that that is a homozygous recessive genotype. It's a lowercase b to get the chocolate color, and then it's blind, so it's a lowercase n. And then it says the black lab is homozygous for both coat color and normal eyes. So that means it's a homozygous dominant black lab. I'm going to pull up the whiteboard again and kind of draw a little area over here to figure this out. So we know that one parent is homozygous dominant for both traits. So capital B, capital N, capital N. The other is homozygous recessive for both traits. So lowercase b, lowercase n. We don't need to do this Punnett square to figure out the genotypes of the offspring since they only have, all of their alleles are the same that they're passing. All the offspring will be BB, capital B, capital N, lowercase n. You can do the Punnett square to figure this out if you would like, or you can just look at the fact that if you have 2 homozygous individuals that are homozygous for both traits, your offspring will be heterozygotes. So it says all offspring would be black with normal vision, but heterozygotes. So capital B, capital N, lowercase n. We can use something called a test cross to determine unknown genotypes, and a test cross is a mating between an individual of unknown genotype and a homozygous recessive individual. The purpose of this is it can reveal the unknown genotype. So the homozygous recessive individual here is our chocolate lab coat color. We know that color is recessive. And then we have a black dog here. We know because it's black, it has at least one dominant capital B allele, but we don't know what the other allele is. It could be another capital B, or it could be a lowercase b. There's no way to know just based on the phenotype of the dog. However, if we complete a test cross with a chocolate lab that's homozygous recessive, based on the frequencies of phenotypes in the offspring, we can determine what the genotype of the other parent is. So if this black lab is homozygous dominant, if it's capital B, all of the offspring would be black from this test cross because all of the offspring would inherit one capital B from this parent. There's only the possibility to inherit a capital B from this parent. So all the offspring would be black, and then they would inherit a lowercase b from the other parent. So if you did this test cross and all your puppies were black, you would know that the black lab parent was likely capital B, capital B. If you did this test cross and ended up with some black lab puppies and some chocolate lab puppies, you would know that the black lab parent has to carry that recessive allele in order to produce chocolate puppies. So you would know that the parent's genotype was a heterozygote, capital B, lowercase b, because some of the offspring would get the capital B from this parent, some of the offspring would get the lowercase b from this parent, and then all the offspring would receive a lowercase b from this parent because those are the only allele options that it has to give. One thing I want to make clear again, this doesn't mean that you would just get two puppies, one black and one chocolate. These are ratios. So one black to one chocolate is a 50-50 ratio. So let's say you had a litter with 8 puppies, you would likely see a ratio close to 50% black and 50% chocolate. Same with this big dihybrid cross here with the 9-3 to 3-1. Those are ratios. So if you did this cross and had a litter of puppies, for the ease of explaining this, we'll say that there are 16 puppies. 9 of them are black, normal vision. 3 of them are black and blind. 3 of them are chocolate with normal vision. And then 1 of them is chocolate and blind. But these are just percentages. So no matter what the litter size was, you would expect to see a similar ratio once you reduced it down. Okay, back to the PowerPoint. So test crosses are really helpful to predict an unknown genotype. You need to just cross that unknown genotype to a homozygous recessive. So a checkpoint question based on that, you use a test cross to determine the genotype of a lab with normal eyes. Half of the offspring are normal and half develop PRA, which means they go blind. What is the genotype of the normal parent? So instead of looking at coat color, we are just looking at eyes now. We have a lab with normal eyes. We don't know if that lab is capital N or a heterozygote capital N lowercase n. We're going to do a test cross, which means we'll cross it with a homozygous recessive, so a lowercase n dog that is blind. And we end up with half the puppies that are normal and half have PRA. Based on that, we know that the parent has to be heterozygous or else none of the puppies would have developed PRA, because if the normal parent was capital N, all the puppies would not have the chance to develop PRA. Okay. Mendel's laws reflect the rules of probability. We're going to talk about 2 rules of probability here. We have the rule of multiplication and the rule of addition. The rule of multiplication can be used when you are predicting which allele from a parent will be passed on to the offspring. So the rule of multiplication calculates the probability of 2 independent events both occurring. So we're using the dime to represent the female eggs and then the pennies to represent the sperm from the male. So both of these individuals are heterozygotes. They're capital B, lowercase b, and then the flip of the coin determines whether this female passes a capital B or a lowercase b onto her offspring. We know that every time you flip a coin, it's a 50-50 chance of landing on heads or tails. So there's one half ratio. 1/2 of the gametes will get the capital B, and then 1/2 of the gametes will get the lowercase b. Same for the male. 50-50 chance. So half of the gametes will get the heads or the capital B, and then half of the gametes will get the tails or the lowercase b. One quick thing about probabilities. So if I flip a coin 4 times and it lands on heads every single time, and I'm about to flip it for the 5th time, is the likelihood of the coin landing on heads higher, lower, or the same based on the fact that for the 4 previous times it landed on heads? Think about that for a second. So the answer to that is that the probability is exactly the same. Each coin toss is an independent event, so it doesn't matter that the 4 coin tosses beforehand were all heads. The slate wipes and starts again each time I flip the coin again. This is how like casinos and gambling gets people, because we tend to be superstitious and we think, oh, like it's done that so many times, like there has to be a pattern, which is not necessarily the case. Probability is exactly the same every time an event happens, no matter what has happened in the past. So every time you flip a coin, it's a 50-50 chance of head or tails. Even if all of the previous 5 tosses have been heads, it doesn't make it any less likely or any more likely to land on heads during the 6th toss. Okay, so the rule of multiplication, we know that there's a half chance that the eggs get the capital B or the eggs get the lowercase b. And then same for the sperm. We then multiply those 2 probabilities to determine the likelihood of them both occurring. So 1/2, which is the probability of the egg inheriting the capital B, times 1/2, which is the probability of the sperm inheriting the capital B. 1/2 times 1/2 gives us 1/4. And we see that in the Punnett square. 1/4 of the offspring have capital B, capital B. You can do the same thing all the way across. It's 1/4, 1/4, 1/4, because it's 1/2 times 1/2, 1/2 times 1/2, so on and so forth. This gives us the same results as a Punnett square would, essentially. So 1/4 of the offspring will be capital B, capital B. 1/4 of the offspring will be capital B, lowercase b, with the capital B from the mom, the lowercase b from the dad. 1/4 of the offspring will be capital B, lowercase b, with the capital B from the dad and the lowercase b from the mom. And then 1/4 of the offspring will be lowercase b, lowercase b. That brings us to the rule of addition. The rule of addition calculates the probability of an event that can occur in alternative ways. In this Punnett square, the event that can occur in alternative ways are these 2 heterozygotes. So both of them are a heterozygote with capital B, lowercase b, but they can occur in 2 different ways. This one occurred with the capital B from the mom and the lowercase b from the dad. This one occurred with the lowercase b from the mom and the capital B from the dad. To calculate the probability of an event that can occur this way or this way, we just add them together. So it's 1/4 plus 1/4. That gives us 2/4, or you can reduce that to 1/2. And we do know that half of the offspring will have this heterozygote genotype, 1:2:1, that ratio that we talked about. Now we're going to talk a little bit about pedigrees. So genetic traits in humans can be tracked through family pedigrees. The inheritance of many human traits follows Mendel's laws, and a family pedigree, which is also just a fancy word for a family tree, can help determine individual genotypes. Typically, in pedigrees, circles represent females and squares represent males. We're going to take a look at a specific pedigree over the next few slides. This pedigree deals with a widow's peak hairline trait, which is when your hair kind of comes to a point, versus a straight hairline trait over here, which is when there's no point, it's just flat across. The straight hairline trait is homozygous recessive, so you need 2 lowercase h's to have the straight hairline trait. The widow's peak hairline trait is dominant, so you just need 1 capital H to have the widow's peak. We have our 1st generation, 2nd generation, and 3rd generation here. In pedigrees, a line like this represents a mating, and then a line vertically represents the children. So Al and Beth had Evelyn, Frank, Gary, and Henry. Those are their children. And then Isabel and Julia are the children of Charles and Debbie. And then Kristen and Libby are the children of Isabel and Henry. So we're going to look at Kristen first. Kristen has a straight hairline, but her parents do not. Remember, the straight hairline is the homozygous recessive trait, lowercase h, lowercase h. So we know, just based on the fact that Kristen has the trait and her parents do not, that the straight hairline must be homozygous recessive. So if we were not told up here that the straight hairline was homozygous recessive, we would be able to deduce that based on the fact that Henry and Isabel do not carry the trait. They're the clear circle and square, and Kristen does. That would mean that it's a homozygous recessive trait. And then a parent with a widow's peak who has a child with a straight hairline. So both Isabel and Henry have the widow's peak. That's the dominant trait. But their child, Kristen, has the straight hairline. That means they both have to carry that recessive allele. They have to carry the lowercase h. So we know that both Henry and Isabel are capital H, lowercase h, based on Kristen's genotype. Another thing to note is that not all genotypes can be determined. Libby over here, we know she's a widow's peak, but widow's peak is the dominant trait, so we don't know if she's capital H, or capital H, lowercase h, and there's no way really to know unless she has some children and the pedigree is extended, because based on her parents' genotypes, she could be capital H, or capital H, lowercase h. So that was a quick look at a pedigree. We have a checkpoint question based on this pedigree. If Libby had a child, which phenotype would allow her to deduce her own genotype for certain? So think about that for a second. I'll give you a second to look back at this. So if Libby's child had a straight hairline, lowercase h, then Libby would know that she herself must be a heterozygote, capital H, lowercase h, or else she wouldn't have been able to pass on that recessive trait, the straight hairline trait, to her child. If she had a child with a widow's peak, that wouldn't really tell us much because she could still be a heterozygote or a homozygote and pass on the capital H. Many inherited traits in humans are controlled by a single gene. That widow's peak or straight hairline that we just saw is an example of this. So are the occurrence of freckles or the occurrence of pigment in your skin and hair versus albinism or albinism. I said that weird. This image just goes to demonstrate that recessive traits are not always less common in the population. I think that's sometimes a misconception. So having freckles is a dominant trait. Having no freckles is a recessive trait. You need two recessive alleles to not have freckles. However, no freckles is more common in the population than having freckles. And then down here, having skin and hair pigment is a dominant trait. Albinism is a recessive trait. Albinism is less common, whereas having pigment is more common in the population. So this just goes to show that recessive traits are not always less common in the population. So just make sure you're aware of that. The genetic disorders listed in the table on the next slide are known to be inherited as dominant or recessive traits controlled by a single gene. So we're going to look at a few examples. We have the disorders here on the left and then the major symptoms on the right. The disorders are grouped by recessive disorders and dominant disorders. So we're going to kind of talk through each of these. We'll talk through the recessive disorders first. Albinism is a recessive disorder. The major symptoms are a lack of pigment in the skin, hair, and eyes. Cystic fibrosis is also a recessive disorder. The major symptoms of that are excess mucus in the lungs, digestive tract, and liver, increased susceptibility to infections, and death in early childhood unless treated. Phenylketonuria or PKU, the major symptoms of that are accumulation of phenylalanine in the blood, lack of normal skin pigment, developmental disabilities. Sickle cell disease, the major symptoms are sickled red blood cells, damage to many tissues. Tay-Sachs disease, the major symptoms are lipid accumulation in brain cells, mental deficiency, blindness, and death in early childhood. And then our dominant disorders, we have achondroplasia, which is dwarfism, Huntington's disease, which is the symptoms are uncontrollable movements, cognitive impairments. It strikes in middle to late middle age. And then finally, we have hypercholesterolemia. The symptoms of that are excess cholesterol in blood, which likely leads to heart disease later on in life. Notice that just in this short list, with the exception of Huntington's disease, the dominant disorders tend to be a little less serious, whereas the recessive disorders, besides albinism, a lot of times result in early death or death at a young age. So generally, we see that recessive disorders are a lot more fatal or severe than dominant disorders. Most people who have recessive disorders are born to normal parents who are both heterozygotes. That is, parents who are carriers of the recessive allele for this disorder, but are phenotypically normal. Whereas for dominant disorders to get passed on, that individual's parent has to have the disease as well and be showing symptoms of it because it's a dominant disorder. So this leads to selective pressure against dominant traits because a lot of times if the disease is serious enough, the individual will die before getting the chance to reproduce, which is why a lot of the dominant disorders are less serious, with the exception of Huntington's disease. But something to note about Huntington's disease is that it strikes in middle age or late middle age. So a lot of times people have already had kids by the time they develop Huntington's disease. So it doesn't impair their ability to reproduce. Whereas these recessive disorders, like is mentioned here, most people who have these recessive disorders, neither of their parents had it. Their parents were just carriers and possibly didn't know that they were carriers. So this is how these recessive traits that can cause some nasty genetic disorders stay in the population because they are recessive traits, and so those alleles are in the population in the heterozygotes. So this shows, we'll say these are two parents who carry the albinism gene. So the capital A is normal, so normal pigment, the lowercase a is the albinism gene. If we have two heterozygotes that have a kid, they have a 25% chance or a 1/4% chance to have an albino child. They have a 50% chance to have a child that's a carrier like themselves of the albinism trait. And then they have a 25% chance of having a child that is completely normal and does not even carry the albinism trait. So this is how recessive genetic diseases and disorders stay in the population, is because they are passed down through carriers. And even if two carriers have a kid, there's only a 25% chance that that kid will be affected. So those traits stay in the population. A checkpoint question. Peter is a 30-year-old man whose father died of Huntington's disease. Neither Peter's mother nor a much older sister shows any sign of Huntington's. What is the probability that Peter has inherited Huntington's disease? So remember, Huntington's disease is a dominant disease and it doesn't strike till middle age. So Peter's father had it. That means he has to have at least one dominant allele for Huntington's disease. It's likely that he probably doesn't have two, because that would mean both of his father's parents had the dominant allele, which is unlikely. So we're going to assume that his father was a heterozygote. So since his father had the disease, there's a half chance that Peter received the gene, or a 1 in 2 chance. The genotype of his sister is irrelevant. She also has a 1 in 2 chance to receive the gene, but her genotype does not affect Peter's in any way. So there is a chance, there's a 50% chance that Peter has inherited Huntington's disease if his dad was a heterozygote. When we're thinking about genetic diseases and disorders, there have been a lot of new technologies that can provide insight into one's own genetic legacy, such as all those genetic tests that we talked about at the beginning of the lecture. There are also a lot of tests you can do if you're thinking about having kids or once someone is actually pregnant. such as carrier screening, fetal testing, fetal imaging, and newborn screening. All of these can provide information for reproductive decisions, but they may also create ethical dilemmas. We're going to talk about two types of fetal testing: amniocentesis and chorionic villus sampling. Both of these allow you to get a karyotype of the chromosomes of the developing embryo. In amniocentesis, a needle actually extracts the amniotic fluid from around the embryo. That amniotic fluid contains some cells from the fetus. Those cells are then cultured for several hours to several weeks, and then after that happens, you can get a karyotype, and then the cultured cells, after several hours, you can do biochemical and genetic tests on those cells. Chorionic villus sampling, or CVS sampling, a needle is inserted into the cervix to actually take some fluid from the placenta and the chorionic villi that connect the mom and the embryo. Those fetal cells, in several hours, you can do biochemical and genetic tests on them, and in several hours, you can also do a karyotype versus several weeks from amniocentesis. So both of those are ways to sample a developing embryo to see if any genetic diseases or disorders are present. We know with karyotyping, you'd be able to see if they have the normal amount of chromosomes or if there's any abnormalities there. That's kind of where we're going to end this lecture. So this video and the previous video, we're talking about Mendel's laws. Now we're going to talk about some genetic variations on Mendel's laws in the next video.